Welcome to the Jack Westin MCAT Podcast with your hosts, Michael Maleka
and Molly Hudash. Hello, everyone, and welcome back to another episode of the Jack Westin MCAT Podcast with your hosts, Mike and Molly. Mike, I'm very enticed by what you're doing here because it is such a beautiful representation of what we will be talking about today and actually for the next couple episodes.
Yeah,
right?
Absolutely.
Since we're going to talk about
Metabolism. Oh, sorry. Yeah, let me do that.
give me enough time for the drum roll.
Did the drum roll? Yeah, I'm sorry,
Okay.
but we're gonna be talking about metabolism over the
There
next
we
few
go.
episodes. And for those of you who aren't watching via video, I was just eating. I was just having a granola bar today. Maybe it's part of the thumbnail. Who knows? But I'm just eating a granola bar, and this is something that we've all done before. I mean, maybe not a granola bar, even though I think most people have probably eaten a granola bar before, but we've all eaten before. I hope. Yes, I can assume that. We've all regularly eat every single day. We're going to be talking about the biological processes that happen after. Like, how does our body use this energy? Because that's what we're really doing, right? When we're eating, all that we're doing is putting energy into the body. This granola bar I have in my hand isn't just seeds and nuts and whatever else is in here, and probably some sugar too, right? It's energy. And our body is going to want to extract energy. This is gold, but our body can't use it quite yet. It needs to extract the gold, extract the value so that we can actually use it in our processes. And that's the process of metabolism, which is extracting that energy and changing it so that we can use it in our own body to power other processes. So I'm very excited to talk about it because we can't cover it all today. We're going to give an overview today of like the processes, and we're going to dive into, I think, probably one of the most tested parts of metabolism on the MCAT.
Yeah, most definitely. I feel like you can't even argue that, right? We're going to talk about an overview. We'll get you introduced to the key players in metabolism, especially catabolic processes, which is the subset of metabolism that is taking a compound and breaking it down, breaking bonds for energy, stripping energy away from a system, versus anabolic reactions are using that excess energy to build something, typically some sort of biomolecule, right? Metabolism really is the sum of both. You've got your anabolic reactions that are powered by your catabolic reactions. That's why you need to eat a granola bar or whatever you're eating, right? It's necessary because we are constantly building things, building new proteins, reproducing our cells, etc., right? We're also not perfectly efficient creatures, right? So even if you're not actively, growing a fetus or a new limb, or if you're not actively getting much taller, you're still undergoing a ton of cellular turnover. Think red blood cells, think skin cells. You need to constantly have energy coming in from your diet, from your surrounding environment to power through catabolism those biosynthetic reactions where you're building new stuff. That would be anabolism. So we'll talk about the major players here, and then yes, we will talk about glycolysis. But really, over the next couple episodes, I want you guys to know that we're going to dive in much deeper into every one of these processes because they're all critical. And I think even if you aren't like testing next week, this stuff is cool. I think it's just
cool.
so cool.
It's super cool. We were nerding out about it. We always talk about what we're gonna talk about before we actually start recording. And this stuff, we were both just sharing how much we love sharing certain aspects of it, certain aspects of the whole pathway that are just super cool that we're both very, very passionate about. So super excited to dive into it.
Yes. So let's start again. We're going to start with an overview, but let's first start just walking through aerobic respiration. What is the point of it, and what are the kind of major processes that we would expect to see as part of aerobic respiration?
And so I think before we even dive in, we have to know what the term aerobic respiration means. It's aerobic means has oxygen. Oxygen is present, and respiration is this idea of breathing. And so this is how our body processes energy or produces energy when we have oxygen. Now, there's a few steps that are involved in aerobic respiration. The first step is glycolysis, right? And we have to think about what's the entry point into aerobic respiration. It's glucose. Glucose is the main thing the MCAT talks about when it comes to breaking down those foods that we eat. It does talk about fats, which we'll get into in another episode, and like a
And
little bit
amino
on
acids
proteins
too.
and a little bit on
Yeah.
amino acids, but carbohydrates is like the main
Yeah.
thing the MCAT loves to talk about when it comes to aerobic respiration. And the main carbohydrate is glucose. So glucose is the entry point, and the goal of glycolysis is to take that glucose and essentially get it ready, right? It's essentially start extracting a little bit of energy, but essentially get it ready for the other extraction processes. Because again, glucose has the energy in its bonds. And so we want to take that energy out. Again, it's a catabolic pathway. We want to break down that glucose to power our anabolic processes. So we break down glucose into a molecule known as pyruvate. And now in that process of breaking down to pyruvate, there's kind of there's kind of some things to keep in mind. Glucose, and I always kind of like when sharing about aerobic metabolism, track your carbons. Glucose has 6 carbons and each pyruvate has 3. And in the process of turning this glucose into 2 pyruvates, you produce some energy molecules. And again, we'll dive into what those energy molecules
Yeah.
are when we dive
More to
into more
come.
more to come.
More to
But
come.
essentially now we have pyruvate. Now, when we have this pyruvate, what's next, Molly? After we've gotten this glycolysis, again, we'll dive into it more later. Where do we go next?
Yeah, so glycolysis is a really important reaction. We're going to go through every single step. This, we're just doing a kind of nice broad overview. So we take pyruvate. Now we've essentially chopped glucose in half. We have 2 halves, which is pyruvate. We're going to oxidize pyruvate into a molecule called acetyl-CoA with the pyruvate dehydrogenase complex, which we'll
EDC.
talk about later, which is like the bridge between glycolysis and the citric acid cycle, which is the next stage, right? And again, in each of these processes, oftentimes we're stripping away energy as we do, but we're taking pyruvate, making acetyl-CoA, which then goes through the TCA cycle or the citric acid cycle or the Krebs cycle, whatever you want to call it, right? Where that acetyl-CoA is going to fuse with oxaloacetate, which is the end product. We're going to go around in a circle and strip off the remaining carbons that originated from glucose, and make a bunch of kind of temporary energy storage forms, right? We alluded to them earlier with redox, right? Since ultimately,
Oh
like
yeah, throwback.
all of aerobic respiration is a
Right.
giant redox reaction where glucose is oxidized into carbon dioxide and then oxygen is reduced into water. We make a bunch of, we call them reduced electron carriers, things like NADH and FADH2. That's going to the electron transport chain where we're essentially going to cache those in for ATP through oxidative phosphorylation. We're going to talk about every one of these steps in a lot of detail.
Yeah.
That is to come because these are really the steps that are tested in the most detail. Okay, aerobic respiration, you want to know the ins and outs. You want to know where in the cell or even in the body, if it's a specific tissue type that has this reaction, where does it happen? When does it happen? Right? What are the major intermediates? What are the major enzymes? Right? How are they regulated if they're regulated, et cetera, right? So there's a lot to know about aerobic respiration. And then we could also talk about some of these pathways that kind of jut off of aerobic respiration, right? Things like glycogenesis and
Genolysis.
Thank
Oh
you.
wait,
Like we didn't
glycogenolysis.
glycogenolysis and
Yeah.
glycogenesis, which is all
Right.
about storing glucose. We could talk about the pentose phosphate pathway, which uses one of the intermediates of glycolysis to actually go on and do some biosynthetic reactions. So anabolic, right? Pathways splitting off of glycolysis. And then there are different ways that we have other macromolecules, like you said, kind of joining this aerobic respiration pathway. So we could talk about how fatty acids and fats can be used to make acetyl-CoA.
Yeah,
Can
beta
then
oxidation, right?
Yes, exactly. And we could also talk about how different amino acids can join this pathway. That is a lot less like certain and specified because we have 20 amino acids.
Yeah, each
Some
of them
of which
enter at different points. Yeah.
Yes, exactly. And I feel like it's important to recognize this. Even glycolysis, which we'll talk about today, can even run backwards. If we want to go pyruvate to glucose, we could do that too. Okay. What we want to be doing here in giving you this overview is recognizing that you have not just 3 processes to know or 4 processes to know. There are more, but they're all interrelated.
That's
They
the key,
all
right?
they all share intermediates. They all share context as well, right? There are processes that we prioritize when we're hungry,
Mhm.
and we therefore have not eaten in a long time. We don't have a lot of energy stored, right? And there are processes that we do when we're fed,
Mhm.
right? And those are kind of big, major ideas that
Mhm.
kind of tie in with regulation.
Right.
But we'll talk more about all of this stuff.
All those things. And I think it's also really cool because I know, I mean, over the years, like we know, like students struggle with metabolism, right?
Yeah.
Metabolism is this idea that is really hard because we know as students always just try to memorize the steps. They just try to
Yes.
memorize all the steps of metabolism and then wonder why they can't answer questions on them. Maybe someone listening right now is feeling that way where they're like, I've memorized glycolysis 100 times. Why can't I answer questions about it? Because I mean, it comes back to this idea that if you're just watching, it's going to be new, but this is something we talk about all the time, which is we have to understand these processes, why these processes are happening, and recognizing they are all connected. A big
Yeah.
trouble people have is like, oh, I'm going to study my glycolysis chapter. Then, okay, that's totally done now. Now, let me study gluconeogenesis now, as if they're like totally two different things, right? And totally unrelated. Absolutely not. All these pathways are interconnected or intertwined. You impact glycolysis, yeah, TCA is going to be impacted because they're all related. Right.
Yeah.
And so this is the idea that we're going to be kind of approaching over these next few episodes. We're going to be really going to be focusing on the why behind these processes. Why is it that this step is important? What is the purpose of, the production of this energy? How does this relate to those other processes? Where do we branch off and why would we branch off? What environmental conditions would we want to do that? We'll be talking about all those. And if you are somebody, you're listening right now, you're like, yes, I have been struggling, with bioenergetics and aerobic like metabolism, like this is definitely where you need to be because we'll be diving into all those things.
Yes.
Mhm.
And I feel like it's really important to recognize that. Like there will be some questions that are like, what is this intermediate of glycolysis? What is the name of it? Or what enzyme produces it? Like there will be some questions occasionally that will hit on your direct knowledge. Usually, if you see a question like that, usually it's glycolysis, which is why
Sure.
we'll take so much time to go over it.
Mhm.
But what we don't realize is how frequently metabolism questions are not actually asking about one direct step.
Mhm.
They're going to mutate a protein or an
Mhm.
enzyme somewhere in that cascade, and they're going to ask you what's going to happen to a
Right.
related process.
Mhm.
That is never something that you will see in a textbook because
Mhm.
it's something that's unique that they're presenting to you, right?
Mhm.
They're going to mess with processes and ask how they will be affected. They'll
Mhm.
maybe upregulate a process and ask what's going to happen. They
Mhm.
may, ask you about connections.
Mhm.
And that's the stuff that I think most of us feel very unprepared for. Okay,
Mhm.
so that's why we're going to set our stage right now with the basics of glycolysis.
Mhm.
And actually, we'll go through a lot more than the basics.
Just the base. I think we're gonna talk. I think everything
It'll
you
be
really need to know about
pretty
glycolysis. Like,
comprehensive on
yeah, yeah,
glycolysis. Yeah.
yeah.
Um, but we're going to start there. And
Mhm.
then over the next couple episodes, we're going to highlight the full process of aerobic respiration that you really need to know, but then also how do other biomolecules come into this? When do we build molecules? When do we break molecules? How is this aerobic respiration process and idea relevant to the human body as a whole?
Mhm.
Right. So it's going to be pretty fun.
Mhm.
And I feel like at this point, we've set our stage.
Mhm.
We're ready to jump in with
Let's
glycolysis.
dive into it. And I know when we were talking about before, we're like, oh, which one do we want to talk about first? And I know, Molly, you love the first parts of glycolysis, which we'll talk about. And I love the second parts or that's going to break it up. And that's how we're going to break it up. But again, let's go back to setting the stage about glycolysis. And I kind of like, when I talked about glycolysis, I realized like, oh, I was going way too much detail. But this is the place to be talking about detail when we first, now that we're talking about it, really focusing on it. So going back to this idea of tracking our carbons, recall, glycolysis starts with glucose. Glucose is the sixth carbon molecule that all the energy is in the bonds. And the goal of glycolysis is kind of a few. First, it's to break that glucose into two equal pyruvates, identical pyruvates. And each one of those pyruvates are 3 carbons. Now, I have 2 3-carbon molecules and I started with 6. Have I lost any carbons in that process? No. So therefore, no CO2 is produced in this process because all the carbons are accounted for. Now, this is kind of I've seen questions of people like, how would I know? Do I have to memorize everything that's producing, every like little thing that's producing glycolysis? No. The carbons are all still there. No CO2 is produced. Done. It's all done and dusted kind of deal. So no carbons are lost. And we also need to know, where does glycolysis happen? So we know it's the goal of taking this glucose, turning it to 2 pyruvates. This happens, though, in the cytoplasm. Now, this is very important that it happens in the cytoplasm. And again, I'm going to steal your thunder about evolutionary processes, because this is actually really evolutionarily relevant when it comes to like where our kind of processes come from. Because we know that other processes that we kind of alluded to, a lot of them happen in the mitochondria. The mitochondria cells do not always have mitochondria. There's this kind of a theory that the mitochondria like was a bacteria that implemented into these into our kind of primordial cells. And so before the mitochondria was there, the idea is that all cells had was glycolysis. So glycolysis is a very conserved process across all cells, really. Like a lot, like I think, again, you can confirm this because you're more into evolutionary. I believe all cells can perform glycolysis, correct?
Mm-hmm.
Yeah, every single cell on the planet can do glycolysis. Not all of them can do the other steps because not all of them have mitochondria. So again, glycolysis is kind of cool evolutionary aspect that, who, surprise, surprise, I'm the one bringing it up today. That is really conserved across all these cells. And what's important is not only that it occurs in the cytoplasm, but doesn't actually require any oxygen. Now, this is kind of an important part that I want to illustrate. Glycolysis is part of aerobic respiration, the first step of aerobic respiration, which we said is a respiration that occurs when we have oxygen, but glycolysis itself does not require any oxygen. Okay, so that's an important part. It happens in the cytoplasm, no oxygen is required. Okay, and so when we talk about glycolysis, again, big picture idea, the idea of glycolysis is that we are trying to produce not only two pyruvates, but we also have other energy molecules that we're producing. Because again, glycolysis isn't just cutting the like glucose into two pyruvates. We also produce a little bit of energy. Not a ton, to be honest, but a little bit, right? And this is going to be important when we talk about anaerobic processes. But net, glycolysis not only produces the 2-pyruvate, also produces 2 ATP. Again, those really high energy molecules that we can use to power those biosynthetic processes, but also 2 high energy electron carriers, which we've discussed before, which are NADH. NADH is produced, and these are very important electron carriers. They themselves, like we can't use them for energy quite yet. They're not like ATP, but they're really important for transporting those electrons to a place where we can actually harness that energy from the electrons, which we'll talk about in another episode. But that's when we're kind of breaking down that glucose, turning it into pyruvate, we also get a little bit of energy out of it. Again, when it can happen in oxygen conditions, but it can also happen in non-oxygen conditions. And that's kind of like the big picture overview. I don't think I missed anything that's really critical there for the big picture. Did I, Molly?
No, I don't think so.
Yeah.
Right.
Okay. Yeah. We set the stage there.
Yeah, we have a 6-carbon glucose becoming a 3-carbon pyruvates with 2 ATP and 2 NADHs. Right. We'll talk about fermentation later, basically how we can allow glycolysis to continue happening if that's all we can do. But let's just keep it with glycolysis first. Let's see if we can understand the process. Glycolysis is also a 10-step reaction. A lot of people will ask, do I need to know all 10 steps? Unfortunately, fortunately, because it's really cool.
Because it's cool.
Yes, right, you do. That being said, in order to help you learn what's going on at each step, learn the intermediates, learn the enzymes, learn the ins and outs of each equation or each reaction of the 10, it's really valuable to split this into phases. Okay, and this is something that I do a little differently personally.
You're unique on this one,
than most. The vast majority of textbooks and schools of thought cut glycolysis into 2 pieces. You have an energy investment phase and an energy payoff phase. The idea of the investment phase is we're taking ATP that we already have and we are using it to make glucose more reactive and easier to split in half. We basically have to bribe glucose to be willing to undergo these reactions. Okay, so we have to use a little ATP to kickstart it. 2 ATP come in, and then in the payoff phase, we're ultimately going to get those 2 back and 2 more, which is why, as you said, the net production of ATP is 2. You use 2 of your own ATP, but you get 4 out. Okay, so every round you produce 2 ATP. Again, you also produce 2 NADH. And this works. Usually people say investment phase is the first 5 reactions and the payoff phase is the last 5. That being said, I think it's hard to imagine how all 5 of the steps of the first 5 steps of glycolysis are energy investment. Because what we see is, and I'll keep it super general because we'll go through each step, we're going to invest in ATP And then we're going to switch the molecule around to make it a little more symmetrical. We're going to invest another ATP. Those three, step 1, 2, and 3, that's energy investment. Step 4 and 5, we're actually going to chop glucose in half. It's not glucose anymore, but the molecule that is made from glucose, chop it in half. We're making 2 different pieces. And then step 5, we're going to make those 2 pieces identical. Okay, so what I see is we have an energy investment phase, which is steps 1, 2, and 3. That's where you actually invest ATP. Then I see this kind of hidden second phase, which is splitting or cutting. Cut the molecule in half and make both halves identical. This is important because we have a lot of enzymes for our 10 steps, and you want to make sure if at step 5 we cut it into step 4, we cut it into 2 different pieces. I don't want to have, 5 separate enzymes that will work on these separate products. It's worth it. Convert those 2 to each other and then use the same enzyme and pathway to break them both down, right? So invest energy, split it apart and make the 2 halves equal, and then gather as much energy as you physically can from those molecules. I know that's not the traditional way of thinking about it, but to me, it helps a lot.
Yeah, no, 100%. And again, I was of the camp, like I've always taught it like, oh, you invest and then you take out. But again, when we were talking about it, I love this idea of the cutting, because you're right. You're not really putting anything in during those 2 steps, but you're not really getting anything out. You're really just cutting them and making them identical. So it's kind of cool, this idea, like just make it its own step, which I think is really, really cool.
I love whenever we, and this is a little behind the scenes insight, whenever we get together, Mike and I have obviously we teach the same material, but we have pretty different ways of approaching it, which is so fun. We have different ways to help students remember and to conceptualize and understand and make things stick. And so it's really fun putting our heads together before we teach of like, I do this weird quirk. And it's like, oh, weird. Really? I like that though. It's cool.
Yeah, I learned so many ways. I'm like, oh yeah, and trust me, when we get to muscles, I never forget the Romeo and Juliet thing you talk about with muscles. It's so wild. But yeah, no, it's and I think it really comes back to like everyone's a different learner at the end of the day as well. And so some people like it one way, some people like another, and that's why like I love, if I was doing this with someone like who was identical teaching style to me, like that's be so boring, right? Because it would just be the exact same. So no, I love this.
Yes, this is fundamentally different than the goal of the splitting phase, which is phase 2 of
glycolysis. Right. Wait, again.
Cut it in half and make the 2 identical? No.
We are
keep separate.
I was about to talk about our shared beards, but that's another thing.
That's an inside joke that should stay hidden. Layer that with time. Okay, you guys. So we've set our groundwork. Again, we're talking about glycolysis, and because it is a process that you need to know not everything about, but like kind of everything about, which again, that's our goal. We're going to walk through it all. We're going to go through every step. There are 10. Okay. It would be really helpful. We are a podcast, so you can't see it, but pull up a diagram, please.
If you can.
You do need to know what the intermediates look like. You do need to know what the intermediates are called. You do need to know what the enzyme is doing to those intermediates. And you also need to know what the enzyme is called. If there's anything produced or used in that step, you also need to know that. Okay, so there's a lot. We're going to narrate our way through, but having a visual will help a lot. Okay, so let's start with our very first step. And this is fun because the first step actually is really special. The first step, we start with glucose. We know that. That was our starting material of glycolysis. We take glucose and we are going to invest energy into it. We're using an ATP in our very first step with a kinase, which is the type of enzyme that will phosphorylate using ATP. And it's named pretty well, right? It's called hexokinase because glucose is a hexose. It has 6 sugars and it's adding a phosphate. So we're going from glucose to glucose 6-phosphate, and this is requiring an ATP molecule, right? So ATP, we're leaving ADP left over. And we are phosphorylating the 6th carbon of glucose, right? It's the one that kind of hangs off the chain. If you can picture what glucose
visualize glucose.
Yeah, it's a 6-carbon molecule and it has a 6-membered ring, but one of the members of that ring is oxygen, right? So you start at the carbon that's bound to 2 oxygens and you go all the way around. There's one carbon that's hanging off that six-membered ring, that's glucose 6-phosphate, or that's glucose 6-carbon. Okay, so hexokinase carries out this first reaction. And this first reaction is actually one that is very special because it has a very special function beyond just energy investment. It actually is very critical to our body's regulation of, let's say, blood glucose, right? Because ultimately, when you think of how glucose enters, right, this is not a very active process, right? Glucose is going to pass into the cell and hexokinase is going to phosphorylate it. The thing about not pumping something somewhere is what you're doing is essentially opening a door. Glucose can come in, it can also come back out. Okay? So if glucose walks through this open door into the cell, right? A door that is specifically glucose shaped, right? It's specific to glucose. It comes in and immediately hexokinase is going to phosphorylate it. Now glucose has a negative charge because of that phosphate, and it can no longer fit back out the door it came in because it's fundamentally different. Glucose versus glucose 6-phosphate sounds similar, but that phosphate group is very negative. And again, it's harder for charged molecules to pass through that membrane, and they won't pass through that same, again, door or that protein that glucose entered through. So hexokinase, yes, it's investing in ATP. Yes, it is changing the 6-carbon of glucose, but it's also trapping glucose in the cell because glucose can no longer leave, right? And this is going to continue to allow glucose to flow in because glucose in terms of concentration is just looking at itself. It's like, hey, we don't have that much glucose in the cell. I should keep coming in. Hexokinase is phosphorylating it, and the glucose on the outside of the cell is like, hey, wait, there's still no glucose. Come in, right? So we're changing it. The reaction itself is pretty simple. We're just phosphorylating that 6-carbon, but the purpose of it goes beyond just a phosphorylation. It has a really important function in the context of the cell. Okay, so that's step 1, that's hexokinase. It is also irreversible, and I'm putting this in quotes because there are reactions that we'll talk about that you truly can't go backwards, right? When we talk about pyruvate dehydrogenase complex that goes from pyruvate to acetyl-CoA, you can't go backwards. There's no way. In this case, this enzyme doesn't usually reverse itself, but that doesn't mean that there's not an enzyme that can. Okay. When we talk about the reverse reaction of glycolysis, which is called gluconeogenesis, we just need a different enzyme. Okay. So this is irreversible in quotes, meaning this enzyme doesn't typically go both ways, but there are others. Okay. It is possible to reverse this reaction. Steps 1, 3, and 10 are the ones that they're going to say are irreversible. But if they were truly irreversible, gluconeogenesis wouldn't exist.
Wouldn't be possible.
Right? So just that's our caveat. Okay. So those are the things we want to know about step 1. After step 1, we get a little bit of a breather, right? Glucose, as we mentioned, is a 6-membered ring. One of them is oxygen and 1 carbon is hanging off, right? It would be really hard. We know we ultimately want to make 2 pyruvates. It would be really hard to kind of slice glucose in half and make symmetrical pieces because the molecule is not symmetrical at all, right? So our next step is we're going to take glucose 6-phosphate and we're actually going to isomerize it with an enzyme called phosphohexose, because you have a phosphorylated 6-carbon sugar, phosphohexose isomerase. Okay, we're going to switch it from glucose 6-phosphate to fructose 6-phosphate. Now those two, there's no new atoms coming in, there's no atoms leaving. You're literally just shuffling atoms around. Okay, so glucose 6-phosphate to fructose 6-phosphate. This is a 5-membered ring, and one of those members is an oxygen. So you actually have a lot more symmetry, right? Obviously you're not chopping the oxygen in half, but you can almost imagine how you can make 2 pretty similar pieces from chopping that fructose 6-phosphate in half. Although, of course, we do have an issue. We only have one phosphate. Yes. So we have phosphohexose isomerase make this fructose 6-phosphate, but we also need an extra phosphate on the other side of that molecule to make fructose 1,6-bisphosphate, because now there's 2 phosphates. We're going to phosphorylate that 1 carbon of what is now fructose. And this is going to use phosphofructo, because it's phosphorylated fructose. Yeah. Kinase. And this is specifically phosphofructokinase 1. We will talk about phosphofructokinase or PFK2 later. It's important with regulation because this is actually the slow step. This is the rate limiting step of glycolysis. It's also a step of glycolysis that has hormonal regulation. Very important. PFK1, critical to know for glycolysis. This is the enzyme to know. You should know the others, but this is the one that's asked about a lot because it does such an important job. Okay, again, it's just a phosphorylation step. In the actual chemistry, we're just taking fructose 6-phosphate and sticking a phosphate from ATP onto the 1 carbon of fructose, right? Or specifically onto the oxygen, right? That's attached to this 1 carbon of fructose. But that step is incredibly important. Okay. To summarize, we had hexokinase that uses an ATP. We had an isomerase, right? Phosphohexose isomerase that just makes it a little more symmetrical. Then we have phosphofructokinase, which uses another ATP, right? In my head, energy investment phase is done. We now have a molecule that is roughly symmetrical and has a lot of stored energy because of those phosphates. We're now ready for what I call phase 2. Maybe I should start calling it phase 1.5 because people say phase 1 and phase 2 is energy payoff.
For like investment and payoff. Yeah, I mean, you could, hey, 1.5, 2. Hey, leave it in the comments which one you like better.
Yeah. I'll refer to them by name, right? We're ready for the splitting reactions, right? Or the chopping reactions or whatever you want to call it, right? So we have this roughly symmetrical fructose 1,6 bisphosphate. Again, fructose has 6 carbons. So 1 and 6 are on the opposite sides of the molecule. And we're going to cut it. Okay. This enzyme that does this, which chops it into 2 3-carbon pieces, is called aldolase, right? And aldolase is going to create 2 pieces that are not actually perfectly identical. They're close though. And the 2 pieces are actually isomers of each other. So we're going to make G3P or glyceraldehyde 3-phosphate. I've also heard in some passages GAP. It's the same thing, glyceraldehyde 3-phosphate. We're going to call it G3P because that's how
G3P fans
learned, right?
Yeah.
We're G3P people, but especially because you leave the numbers in every other designation and abbreviation in glycolysis. G3P, but that's the same, glyceraldehyde 3-phosphate. That's one. And the other one is called dihydroxyacetone phosphate or DHAP. These are the 2 pieces that are made. But again, I don't want to have 2 completely separate pathways, one that metabolizes DHAP to pyruvate and one that metabolizes G3P to pyruvate. That would require different enzymes and different regulation. Why not? Because these 2 are isomers of each other, just take one of them and just convert it to the other. Okay. And that's what we do, right? We use an enzyme called triose because it's a 3-carbon molecule, phosphate because they're both phosphorylated, isomerase because it's converting between isomers. So this enzyme TPI or triose phosphate isomerase is going to convert DHAP to G3P. Okay. And we actually have a really interesting passage on this on our website. It's not, you don't have to worry about it, but it's actually why they do that. It's really interesting because actually DHAP is more stable.
Yeah, I know.
More stable.
Right. I actually know the exact passage you're talking about.
Yeah.
And it's funny because we have hundreds. And it's funny we have hundreds of
literally.
passages. I know the exact one you're talking about. And which is, I remember reading that and thinking, I didn't know that.
I didn't know that. Yeah. It's not required knowledge. It's just cool. A lot of our passages, and again, they're free on our website, do really cool things like that. And the MCAT will too. But G3P, DHAP, they're isomers of each other. We're going to take DHAP, convert it to G3P with triose phosphate isomerase, and then we now have done kind of half of what we needed to do, right? We've invested energy to convince this glucose molecule to react enough that we can strip energy from it. And we've cut it into 2 pieces. Both of those 2 pieces will end up to be pyruvate. That being said, we're 2 ATP down. Right? And we haven't gotten anything useful out of it yet.
Right.
So take us home, Mike.
Yeah.
Let's
5 steps.
Payoff phase
Yeah,
now.
let's talk payoff phase. And I think it's cool. And I think it's really, really cool before we move on to the payoff phase to think about the enzyme names. You've been really intentional about the enzyme names. I see way too often people just try to rote memorize all of these enzyme names. Like, notice how, like, anyone who's listening, when Molly was saying every single enzyme, there's a really great reason they're named the way they are, except aldolase. I have no idea what aldolase is called aldolase. I mean, maybe you do. I would love to hear it, but I
I
think
mean, and like aldol condensation. I'm like, what are we
it's
talking
like
about?
cunning,
Why?
but I just think like they are all named so well. Hexokinase, the mutase, the triose phosphate
Yeah.
isomerase. They're all named so
Isomerases.
well, like except aldolase. But this is one thing that's really important, especially for all of these pathways. The enzymes, like 99% of the time, are named really intentionally. That if you don't
Yeah.
have, if you, as long as you know the process, the enzyme name is kind of really straightforward. It tells you just exactly what's happening. And this is not an exception for the payoff phase. Now, super
Mhm.
important to think about in the payoff phase, we are done investing ATP. There's now no ATP being invested at all. All we're doing now is getting payoff. It's all payoff now. And
Mhm.
so right now we have 2G3P. But G3P, well, okay, has that phosphate, it's got a negative charge on that phosphate. We want to make it a little bit more unstable. Because again, we want essentially to get it ready to get the payoff. Again, we kind of want to destabilize it a bit so that when we're ready to get that payoff from it, it's really readily. Because if it's stable, there's not a lot of energy to take from it. So we want to destabilize it. Well, I'm trying to think, okay, if I'm the body, how am I going to destabilize something with a negative charge? Put another negative on it. That's what I would do as the body, right? I would put another negative because two negatives don't like being next to each other. And what's a great molecule with negatives on it? Phosphate, right? Phosphate has a negative. So me as the body, I'm going to add another phosphate to glyceraldehyde 3-phosphate. But here is a really important note. This is
Yes.
step
Really
6,
important and
right?
weird.
Right? We just said no ATP is being used now, and this is no exception. We are grabbing phosphate essentially from the environment, not from ATP, to stick it onto our glyceraldehyde 3-phosphate. These phosphates are not coming from ATP. We are done investing ATP. These are just inorganic phosphates from the environment. They're still negative, and once we stick it on our molecule, right, it'll be now have two negatives, being really unstable, but not from ATP. But if I'm trying to think of like favorability, right?
Mhm.
Does that sound very favorable to stick a negative on top of another negative?
Nuh-uh.
Absolutely not. This is very energetically unfavorable or non-spontaneous. It doesn't want to happen. But Molly, remind me, when we were talking about thermodynamics, how do we make non-spontaneous things happen?
Well, we have to have an investment of energy from somewhere, but
Right.
if not ATP, then where?
We've got to couple it to something else. And this is where I love that we've done so much work to get to this point. This is where we couple it to a redox reaction. And I think the big hint here is the enzyme name that does this whole process of adding the phosphate and coupling to the redox reaction is glyceraldehyde 3-phosphate dehydrogenase. Right. And so that name dehydrogenase, you see that enzyme name dehydrogenase, what should you think of?
Oxidation.
Redox reaction is happening. So we know a redox reaction is happening. What's great, whenever you see the word dehydrogenase in any metabolism, in any case of metabolism, the immediate molecule you should think of is NADH. That's like the first thing that comes to mind is NADH. That's what happens. We take an NAD plus in the environment. Again, keep this NAD plus in mind when we for a little bit later, but we take this NAD plus, and as a result, we couple that reaction, we reduce it to NADH. So we reduce NAD plus to NADH and we oxidize glyceraldehyde 3-phosphate. So there's this redox reaction, which is very favorable. This very favorable redox reaction is what drives the addition of the phosphate to glyceraldehyde 3-phosphate, forming 1,3-bis phosphoglycerate. Again, notice because now we have two phosphates on it. So again, in summary, we just added a second phosphate to G3P. We've done this twice now. We added a second phosphate to G3P, did not come from ATP, and we've essentially coupled that to a redox reaction. So we now have gotten kind of the first little payoff from glycolysis, which is that NADH that will be used later. But now we have this really unstable 1,3-bisphosphoglycerate, right? And now we're like, okay, it's really unstable. Let's get some payoff from that. Now, this is where the next enzyme comes in, which again, it's named very interestingly, which is phosphoglycerate kinase. Now, the term kinase here is very important because when you think kinase before, when we talked about the first phases, kinases take ATP, take a phosphate, add it to something. Remember, this is the payoff phase. We are not investing any ATP. We are taking out ATP. So whenever we see kinase in the payoff phase, think it's doing the exact opposite. It's not taking a phosphate from ATP, it's adding a phosphate to ATP. So phosphoglycerate kinase isn't using an ATP, it's producing an ATP. Remember that 1,3-bisphosphoglycerate has 2 phosphates on it. What's phosphoglycerate kinase going to do is going to take one of those phosphates and stick it on an ATP, right? And this formation of ATP is what we call substrate level phosphorylation, right? Where we have now coupled, right? Because ADP to ATP, not very favorable. But the cleaving of the phosphate from 1,3-bisphosphoglycerate is favorable. Another coupled reaction where we've now produced the first ATP. So now if I'm keeping track now, I now have produced, by taking that phosphate off, producing the ATP, I now have 3 phosphoglycerate. We've taken one of the phosphates. Now there's just 1 phosphate and it's on the 3rd carbon. So if I'm keeping track of what's happened, I have 1 NADH produced and 1 ATP produced. Again, I'm just focusing on one branch right now. We'll talk about like the second branch in a second. So we're just going through one branch right now. So now we have 1,3-phosphoglycerate. Okay, but we kind of taken the energy, we've kind of stabilized it by taking that negative charge away. So now, okay, this 3-phosphoglycerate, I want to make it a little bit more unstable again. Because again, that's how we get energy. We take unstable molecules and extract energy from those molecules. So I want to change it up. I want to mutate it a little bit to make it a little bit more unstable. So the next step is what is catalyzed by the enzyme phosphoglycerate mutase. Again, whenever you see a mutase, just think, oh, I'm just changing things up. With phosphoglycerate mutase, there's a phosphate and a hydroxyl group. We're just swapping those 2 on it. But what's essentially doing is bring that phosphate closer to a carboxylic acid group on it, bringing negative charges closer together. Again, destabilizing the molecule again. So we're destabilizing the molecule, once again, forming 2-phosphoglycerate. Again, we've just brought that phosphate closer to another negative charge, again, making it more unstable. We have one more step, and this is where we actually take water out of the molecule, forming a double bond known as enolase. And so again, this is just further serves the purpose of destabilizing the molecule, getting it ready for that last step. And this 2-phosphoglycerate and undergoing that dehydration reaction by enolase forms a really important molecule known as PEP, or phosphoenol pyruvate. Phosphoenol pyruvate has this negative charge of the carboxylic acid right next to a phosphate. Very, very unstable, this PEP molecule. And that's why now we have this super unstable molecule. Now we get the payoff once again. Let's get that. And this is where the really critical enzyme, pyruvate kinase, comes in. That 10th step, that really important enzyme, right? And there's a lot to talk about with pyruvate kinase, but let's kind of talk about what we've known before. Remember, we are in the payoff phase. But we see the term kinase in the payoff phase doesn't mean we're using ATP. Again, we are not using ATP. We are getting ATP. So what it's going to do, it's going to take that last phosphate on phosphoenol pyruvate, because again, it's still at that phosphate. It's going to take it and stick it onto ATP. This is now our second ATP. We had the NADH produced, then from the phosphoglycerate kinase produced the first ATP. Now we have our second ATP produced from pyruvate kinase, producing our pyruvate, right? And this is really important. We have now produced the pyruvate, and this step, phosphoenol pyruvate to pyruvate, is the 3rd irreversible step, right? It does not go backwards, ideally, like essentially, yes, but that we have, like you said, with a little asterisk there.
That enzyme doesn't
that
usually go
enzyme
backwards.
can't go
Other
backwards.
enzymes can.
Can
Yes.
go backwards in that step. But that process is biologically irreversible. We have now gotten to pyruvate and we've finished. But remember, that was just 1 G3P. If we were to like talk about like what's being produced so far, I have 1 NADH, 2 ATP, and 1 pyruvate. Remember though, we produced 2 G3Ps before. There were 2 of those that were identical. So whatever we just produced, that happened twice. So we're not just producing 1 NADH, 2 ATPs, and 1 pyruvate. No, we have 2 NADH, 4 ATP, and 2 pyruvates. We've just doubled everything because this whole process that I've just walked through happens twice because we had 2 identical G3Ps. And so in terms of the payoff, we now have those 2 pyruvates, 4 ATP, and 2 NADH. But remember, when we talk about our net production, we can't forget we did a little bit of investment right at the beginning during when that Molly was describing. And so when we talk about net, we have to kind of subtract those 2 ATPs. So net, we've produced 2 ATP, 2 NADH, and those 2 pyruvates. Whew, that was quite a bit. But
I know. I love it.
Yeah.
It's a very complicated Well, it's a very long
This
process.
is a long process, but I don't think I think every step is
It's
very intentional. There's a reason for
yes.
every step, right?
Yes.
Uh-huh.
And I feel like it's important to keep in mind that this process, as you mentioned, in terms of evolutionary biology, developed early, a
Mm.
very long time ago,
Mhm.
right? Very, very, very long time ago, to the point where it's pretty much conserved across all living cells, right? Very long time ago. And weirdly, it hasn't changed that much.
No, really.
It hasn't changed that much because it's very intentional. Every step does something specific that helps it
Mhm.
achieve its goal, right? Again, to summarize the ins and the outs, we
Mhm.
had glucose, which is C6H12O6, right? Cool, right? We have glucose itself,
Mhm.
and we are breaking that in half to make two pyruvates that are equal, right?
Mhm.
Two equal halves. As we said, we needed 2 ATP to get it started, but we ended up
Mhm.
making 4. So we
Mhm.
also make 2 ATP net production. You'll hear
Mhm.
that a lot,
Mhm.
right? You don't count the stuff that you had to put in. You got it back out. Great. You broke even. We had net production of 2 and 2 NADH. That NADH is really useful, right? And we
It's
can take
interesting
that
that, yeah.
NADH, the whole
Mhm.
point of doing that, right?
Mhm.
I know we've talked about redox in the past. The whole point of holding on to those electrons is to use them in the electron transport chain.
Mhm.
Okay. That being said, and this is an important idea, if we don't have oxygen, which, as
Mhm.
we'll talk about, is going to be the terminal acceptor of those electrons, keep in mind in the net process of aerobic respiration, oxygen is going to be reduced into water. If we don't have
Mhm.
oxygen, ETC is not going to happen. Okay,
Mhm.
we're not going to have those electrons used in the electron
Mhm.
transport chain, and therefore NADH is not going anywhere.
Mm.
If you want glycolysis, let's say you don't have oxygen, you can only go through glycolysis, and for each glucose, you only get 2 ATP. That's it.
Mhm.
It's
Mhm.
very limited in how much energy it can produce. So if you need to survive as a human being, you need a lot of ATP. You're going to have to metabolize a ton of glucose with glycolysis. You're going to run out of NAD plus really quickly.
Yeah, remember that step 6. It was really, really important to take the NAD plus, couple it to that addition of the phosphate. You don't have any NAD plus, that can't happen.
Yeah, and you don't get any of the payoff, right?
Mhm.
So it's very important, if we don't have any oxygen, right, that rather than proceeding through aerobic respiration, and we
Mhm.
take that pyruvate, we oxidize it, we send it through the TCA cycle, we take all those electron carriers like NADH and FADH2, we shuttle it to the ETC to make ATP. None of that is going to happen. I
Mhm.
need to get rid of NADH, and I need to
Mhm.
make or regenerate NAD plus so I can keep just bustling my way through glycolysis to make a
Mhm.
little ATP per glucose.
Mhm.
This is interesting in evolutionary biology as well, because different organisms do this differently. But
Mhm.
for most, we take pyruvate, which at this point, pyruvate's not useful to us. We
Mhm.
can't further oxidize it to
Mhm.
carbon dioxide. We can't, like the reduced electron carriers, we're already out of
Mhm.
NAD plus. Like it's not productive, right? Pyruvate is kind of a waste product if all you're going to
Mhm.
do is glycolysis.
Mhm.
We have NAD plus. What we're going to do is actually, for most of us, humans included, we're going to take that hydrogen, that proton and electron, and we're actually just going to use an enzyme called lactate dehydrogenase to put that electron and that proton onto pyruvate and make a product called lactic
Lactate, yeah.
acid if it's protonated. So we're taking NADH and we're taking pyruvate, and we actually have a redox reaction occurring that
Mhm.
will lead to NAD plus, which now can run back to step 6 of
Mhm.
glycolysis, keep glycolysis going. And we also have now this waste product called lactic acid.
Mhm.
And this is a byproduct for us. If you think about the tissues that might not have oxygen in the human body, we breathe oxygen, right? This is
Of
not
course.
ideal.
Wait, what?
I know, right? I
What?
know. We talked about the cardiovascular and respiratory system, so
Oh,
hopefully
right.
that's
I
not a
forgot. Right.
yeah.
Maybe I should go rewatch those episodes.
Maybe you should. That was a while ago.
Yeah.
Most often, this happens in cells that don't have mitochondria at all,
Mhm.
like a red blood cell,
Right.
or it's happening in cells that are undergoing such extreme ATP demand that we just don't have enough time, right? You go through glycolysis because you use up all your oxygen, right? You're using it so fast. You're using oxygen so fast that you run out and you just have to use glycolysis to keep powering it. This is most often when you work out. If you're doing anaerobic exercise. And I know we both have a fun fact that we wanted to share. Most people, and of course we both go to the gym, so we both have heard this before. Most people think that soreness comes from lactic acid production in your muscles, right? They think that that's what's causing the soreness, and it's not.
Totally untrue. Like I swear, like every gym teacher, like when I was in high school, was like, "Oh yeah, stretch to get the lactic acid out of your muscles," and I'm like, and I was like, "Oh, I believe that so much." But as soon as I actually learned, like, there's actually like literature support, like not related, like not really at all, right? This
Not
like
not to the soreness.
not to the soreness, right? Like the burning, right?
The burning.
Sure,
Oh my
like
God,
that
my
burning.
arms are on fire doing that
That's
last
lactic
bicep curl.
acid, right?
That's
Yeah.
lactic acid.
But like the soreness you experience like a day or 2 days after, like lactic acid's already drained out of the muscles. Like it drains within like I think what is it, 12 to 24 hours. Like
Pretty
when you
quick.
have by the time like a day, 2 days from now, there's no lactic acid. That's just because you've torn your muscles and now they're in the process of regenerating and healing. And so yeah, fun fact, that's a myth. Lactic acid, not the reason for soreness at all.
It's actually because of tiny microscopic tears in the sarcomeres of your muscle,
Mhm.
right? The little contractile unit of your muscle. You are literally destroying your muscles so they grow back stronger, which is kind of cool. Don't destroy them so much that you have a tear or something
Sure.
major, but
Like a
tear them
tendon
down
tear,
just
like
enough that they come back
Yeah,
closer.
eccentric movements, great for tearing. Way better than concentric movements, by the way.
Okay, this is not a gym class.
Okay, yeah, sorry,
Mr.
yeah,
Personal
just
trainer. My goodness, Mike, come on.
my bad, sorry, my bad.
MCAT.
Yes, MCAT. Coming back to MCAT stuff, yes.
Coming back to MCAT.
But
One thing that I do want to say though with lactic acid, that's really cool. Yes, it causes soreness in your, or not soreness. I almost said that. No, it causes burning in your muscles, but yes, it also drains away fast. The cool thing though is we actually can use that lactic acid or that lactate that's produced. It's not like we've just completely thrown it away, right? What you actually can do, and it's a process called the Cori cycle, lactic acid can flow from areas of buildup in your body, flow around in your bloodstream until oxygen is restored, and it can then re-enter cells and go under the reverse process to reform pyruvate and then go to make ATP. Okay, so just so you know, lactate is not just a throwaway molecule, get rid of it. It can circulate and then ultimately be reformed into pyruvate and then sent right back through so we can get the rest of the energy stored from it, right? Because there's a lot of value still in that molecule. Also, just because it's cool, if anyone has ever had an alcoholic beverage before, that's because certain ethanol producing organisms like yeast actually have an extra enzyme that they take lactic acid and go to get rid of a carbon dioxide and make ethanol.
right.
So, kind
Amazing.
of cool.
Another kind of cool
So
process.
that's fermentation as well.
We don't have that enzyme.
Yes.
So
Do you
we're not
imagine if we did?
I'm pretty sure there are conditions that actually
Every
like
time you eat, you would just
you would
be
just get
drunk.
completely eat and then have like maybe or even exercise because it has to be like anaerobic. So maybe like if I exercise,
Yeah,
you just
that's
get
true. That's true. Yeah.
absolutely lit. But but yeah, no, but
This would be so dangerous.
that would be wildly dangerous.
Wildly, yes.
But you know, coming back to like the MCAT content focus kind of topics. But yes, this idea of fermentation really kind of reiterating the problem that you have if you're just constantly doing anaerobic metabolism is that running out of NAD+. The goal of that fermentation, that lactic acid fermentation, is to take the NADH that you're producing to get that NAD+ back so you can keep glycolysis going, keep on producing energy for the cell. But this is an important part to talk about when you know we're talking about glycolysis and this, you know, environmental conditions that might change glycolysis, because regulation is an important part. We talked about homeostasis. We had a whole series on like keeping the body the same. Glycolysis is the same way, because you can imagine we don't always want glycolysis going full steam ahead. There are times in environmental conditions where we don't want maybe glycolysis to be going full steam ahead producing pyruvate. Maybe we want to do something else with that glucose or glucose 6-phosphate. So what are some of those regulatory molecules that might change where glycolysis goes?
Yeah, and I love that we approach it. We first set the groundwork. Again, like you said, that intermediate of glycolysis after step 1, glucose 6-phosphate, which again has just been trapped inside the cell by hexokinase. it can then go to storage,
Mhm.
right, as glycogen through glycogenesis, right? It can also go to the pyruvate or the pentose phosphate pathway. So we have ways to build and store or build glycogen, which is essentially a glucose storage form. We also have ways to go and use that to make something else, right? Of course, we can also proceed through glycolysis and use that
Sure.
for energy. We do not want this to go constantly. We don't want this process to constantly be going. And also, as we alluded to early, sometimes we're fed and we have all this energy, right? Our body is in a very nice fed energy, excess stage, in which case, use it. Use the energy for stuff, right? If we have a ton of glucose, great, right? If you have a cell that needs repair, go ahead and repair it. If you want to divide, go ahead and divide, right? We have so much food. Go ahead, proceed through glycolysis, make ATP, make biomolecules through the pentose phosphate pathway, right? You're fed, have more glycolysis. That being said, if you're really hungry, you haven't eaten in a long time, your body's in high stress mode, right? Like, I don't know when my next meal, I don't know when I'll get my next meal. I don't know how long I'll have to survive with the glucose stores that I have. You are in like full on lockdown mode. You do not want cells to repair themselves right now. You don't want cells to divide right now. The context of the human body plays a really significant role in regulating glycolysis and the other metabolic pathways that we have. In the case that we are in a fasting state, which is the language we use, if we're hungry, we haven't eaten in a while, we want to conserve, shut down anything that's non-essential, and make sure if we have stuff stored for a rainy day that we mobilize that. Okay, so when we talk about glycolysis regulation, we could talk about cellular regulators, like the conditions of the cell that are impacting how much glycolysis is occurring, but we could also talk about system-wide regulators of glycolysis. And the fun fact is most of these regulators all impact my enzyme PFK1, right?
You love this enzyme. Yeah, that's a really important regulatory step. And also we call it the committed step because like you were saying before, G6P, a lot of people say like, oh, as soon as you do hexokinase, oh, now you're doing glycolysis. But
it's trapped, it's not committed.
Trapped, exactly right. It's trapped in the cell, but like you just like really well put, we can go to pentose phosphate pathway. We can keep on going to glycolysis. We can go to make glycogen. It's not committed to anything after hexokinase.
Yeah.
Right. But it becomes committed to glycolysis, like essentially trapped in glycolysis after that really important PFK step. And that's why a lot of regulation of glycolysis happens at that PFK step. Of course, it also happens at those other steps, hexokinase and pyruvate kinase, but a lot of that main regulations happens at that really important committed step of glycolysis, which is PFK1.
Yes. Yeah. And I think the regulators, people get really worried about it. They try and memorize it all. They make sense.
It's like, common. Yeah.
Like, what are the general regulators? I want to talk about my friend PFK.
You can talk about your favorite.
What are the general ones?
Sure. And a lot of these regulators regulate like all the steps of metabolism are regulated by these things because it kind of makes sense. Remember, the goal of glycolysis is to essentially downstream make energy, like extract the energy out of glucose. But imagine if I have a ton of energy in the cell, do I need to be going full steam ahead with glycolysis? Absolutely not. So a great inhibitor of glycolysis is ATP, right? A downstream product saying, I have so much of me, stop making more. We're done. Again, very homeostatic. Wait a minute, what does that kind of remind you of?
Homeostasis.
Homeostasis and negative feedback, right? This idea that a downstream product is inhibiting those upstream processes. Yeah, ATP actually goes and inhibits phosphofructokinase 1 and stops, like, we don't need more pyruvate. Let's use this energy instead to store it, or maybe let's create things for like nucleic acids in the pentose phosphate pathway. Let's do other things with it. But a great activator of phosphofructokinase is a condition where we have little energy. Imagine if my cell doesn't have much energy, it maybe wants to produce more. So ADP, AMP very much activates phosphofructokinase because it's saying, hey, in the cell, I don't have any energy.
Low, yeah.
Please produce more energy. And that's where the goal is to extract the energy. And so these inhibitors, ATP and ADP, it sort of just makes sense why they impact glycolysis. But they also, ATP, AMP, ADP also impact legitimately every other aerobic
Yeah.
pathway. And so as long as you know the goal of the pathway, it just makes sense what inhibits it.
Yes, it
Mhm.
definitely does.
Mhm.
And so ATP, AMP, and ADP all regulate, like you said, most of the regulatory steps of glycolysis,
Right.
and aerobic respiration as a whole.
Mhm.
And it makes sense.
Mhm.
The one that didn't make sense,
Free.
and I crack up. I told Mike this earlier. The first time that I heard of PFK2
Mhm.
or fructose 2,6 bisphosphate, I
Yeah, no.
actually thought they were typos.
Right.
Uh,
So to know, different from the fructose 1,6 bisphosphate that we just talked about in glycolysis, right?
yes.
Yeah.
So
Mhm.
that's the difference, right? So PFK1, which is our commitment step, that is critical to control. And you, Mike, you've talked about the intracellular regulators, which is
Mhm.
ATP, AMP, ADP. Those are the
Mhm.
ones within the cell that tell you how much energy is within my cell. But again, we need to know on an organismal level, how
Mhm.
much glucose do I have, right?
Mhm.
We need to talk about hormonal control too, right?
Mhm.
We have hormones that are released, right, by our pancreas that tell us we have a very high blood glucose, we've just eaten, and
Mhm.
we have a very low blood glucose, we haven't eaten in a while, right? Insulin is going to be released after we eat, right? And it's telling the cell, take in glucose. We've got a ton of glucose. Make sure you eat, right?
Mhm.
It's take it in and use it for something, whether you use it for ATP, whether you use it to store, whether you use it for, biomolecule synthesis. Just take it in because we've got a bunch. It's not
Mhm.
healthy for glucose to hang out in your bloodstream for a long time. So
Mhm.
insulin, when it's present, is telling your cell, take in glucose and use it for something, right? Insulin is going to be an upregulator of glycolysis. It's also, if you're not going to use it for glycolysis, might as well store it, right?
Right.
Glycogenesis, which is synthesizing glycogen, you might as well store it. We have a bunch. And also glycolysis. Again, make ATP, use it for stuff. If you need repairs, if you need
So
this or
take it.
that, do it.
Take it.
Glucagon is going to do the opposite. It's going to
Mm.
promote glycogenolysis to mobilize glucose from our storage, right, which is glycogen, and it's going to inhibit glycolysis. That being said, this is not direct.
Mm.
Glucagon and insulin do not directly come in and halt glycolysis. They
Mhm.
don't have a direct impact themselves. What they do is send a messenger.
Mm.
And the messenger is this really cool enzyme called PFK2.
Mm.
Now, this is very similar in name to PFK1. Again, when I first read it in a textbook, I thought it was a typo. I'm
Mhm.
like, that's so silly, you guys. PFK1 is the important rate limiting
Right.
step.
Right.
Um, PFK2 is an enzyme that, in one form, right? It's actually a bifunctional enzyme, which is cool. But
Mhm.
PFK2 in its normal form creates fructose 2,6 bisphosphate
Mhm.
from fructose 6 phosphate.
Mhm.
That is, it sounds like a competing reaction. PFK1 wants to make fructose 1,6 bisphosphate. This wants to make fructose 2,6 bisphosphate. We are pulling from the same pool of fructose 6 phosphate, and we
Mhm.
have got a side reaction.
Mhm.
If PFK2 is active, we're going to
Mhm.
make this fructose 2,6 bisphosphate.
Mhm.
And if this is present, even at a small concentration, it is going to turn PFK up 100
A million
levels.
percent. Like
It
it's
is,
like on the gas kind of deal.
yeah,
Mhm.
exactly. It's forcing it to step on the gas. PFK1, as soon as F26BP gets close, it is upregulated
Right.
to such an extreme degree, right? And you have so much glycolysis happening. So people get confused because they see PFK2 as this side reaction. It's stealing some of the fructose 6 phosphate that would normally be used for glycolysis. That must be bad. No, even if you only have a handful of fructose 2,6 bisphosphate molecules made in that side reaction,
Mhm.
PFK1
Slant.
is very sensitive to
Right.
it and will immediately be upregulated and
Mhm.
then monopolize the fructose 6
Sure.
phosphate.
Mhm.
So PFK2 is going to be activated by insulin. Because
Mhm.
again, insulin is telling the body we have tons of glucose, go ahead and use it. Make ATP, do whatever you need to do. We've
Mhm.
got tons of energy. Insulin will activate PFK2, and
Mhm.
therefore fructose 2,6 bisphosphate will be produced. That will activate PFK1. That's how it impacts glycolysis.
Mhm.
if insulin is no longer there, PFK2 switches to its reverse format, right? Which is fructose 2,6-bisphosphatase.
Haze, right? That term, right?
Yeah, it's a bifunctional enzyme. So it can phosphorylate fructose 6-phosphate into fructose 2,6-bisphosphate. Or if it stops hearing the message from insulin that we've got lots of glucose, it's going to switch gears and start destroying what it just produced. As soon as insulin stops whispering, go through glycolysis, it's going to destroy that fructose 2,6-bisphosphate, remake fructose 6-phosphate. And now what's important is PFK1 loses that gas, right? It loses the upregulation. And so it's going to move slower, right? If you don't have insulin around, which of course, if you don't have insulin, glucagon is probably present. Glycolysis is going to be much slower. It's not that it won't happen at all. You know, your cells still need energy to live, but to a much smaller degree. It's really just what is necessary for the cell to exist. And I love talking about that. I love the organism-wide regulation. I think it makes sense to think of the individual regulators, ATP, AMP, ADP. That makes sense. But
also like the large scale too, the big scale, right? And I love, again, this is a callback to some of those. We've talked about our endocrine system before and how it maintains the homeostasis. So if you're like, wait a minute, she just brought up these hormones, glucagon, insulin, so they were from the pancreas. What's that about? Go watch that episode. Go watch our endocrine episode. We kind of broke down kind of the major hormones to know if you haven't seen it. And again, this is kind of like what we just kind of talked about. Like, if you understand what we've discussed today in terms of glycolysis, like I don't think there's anything else you need to know about glycolysis, at least in terms of a process on its own, right?
Not that I could think of. I mean, every cell undergoes glycolysis. It happens in the cytoplasm. You know the net reaction, you know the enzymes, you know the individual intermediates, you know what the enzymes are doing, you know the broader phases, you know the regulation. I can't think of anything else you need to know about it.
And I think this is a great place to kind of build off for next episode, because we're going to kind of really kind of build off into this next processes, because this all kind of comes, like we've mentioned at the beginning of today's episode, this is all super interconnected. Like the next steps, PDC, TCA, like you've already kind of even hinted at some of those branching processes that we'll go into like much more later, right? It's all related. And so having this really foundational understanding and then building up from here, I think is necessary when like learning about metabolism.
I agree. So like we mentioned, this is just the first episode of our metabolism series. We have covered so much, probably the highest yield stuff. Although I actually think the pyruvate dehydrogenase complex, for the fact that it's seen as one step, it's actually a complex, but for the fact that it's seen as like one step, one reaction, I actually think that is equally high yield. It's tested pretty frequently as glycolysis.
It gets forgotten about all the time.
It gets forgotten about, but it's really important.
It's really important. And so we make sure to give it its love next episode.
Yes, exactly. So we're going to round out aerobic respiration first, and then we're going to talk about some of these other metabolic processes that we've alluded to. And we hope that as you guys are listening to this metabolism mini-series, you are trying to picture how these processes relate to each other. Okay, everyone can Google glycolysis and get the 10 steps. That's not what I mean. What I'm talking about is making a sort of mental map that shows how glycolysis and gluconeogenesis or glycogenesis or glycogenolysis or pentose phosphate pathway, right? How all of these processes interact. That's what we're going to be describing over the next couple episodes here. So if you liked it, make sure you stay tuned for the next few and make a little visual and get practice.
Get practice. On our website, plenty of practice questions and passages focused just on metabolism. So highly recommend checking those out, and totally free. So, I mean, like, what do you have to lose? And so thank you so much for joining us today. And Molly, what do we say normally to end off an episode?
Well, we usually awkwardly ramble of have a good rest of your day or night or week or weekend or month or whenever we find you. And then, of course, we'll tell you, thank you for listening. Happy studying.
Happy studying.
We'll catch you in the next one.
Bye, everyone.
Bye, everyone.